Malignant mesothelioma is an aggressive cancer that forms in the lining of the lungs, pleura, abdomen, peritoneum, or heart, pericardium. It is usually caused by exposure to asbestos. Due to the often late stage of diagnosis and resistance to chemotherapy and radiation therapy, mesothelioma has a very poor prognosis with a median survival of only 9 to 12 months. Immunotherapy using genetically engineered chimeric antigen receptor (CAR) T cells holds promise to improve outcomes for mesothelioma patients. CAR-T therapy genetically modifies a patient's own T cells to target specific antigens on tumor cells. Here is a review of the key ongoing clinical trials researching CAR-T therapy for mesothelioma. One of the most advanced studies is a Phase 1/2A trial at the University of Pennsylvania testing CAR-T cells targeting mesothelin, a protein highly expressed on mesothelioma cells, NCT03907852. Mesothelin is an ideal target as it is expressed on up to 90% of mesothelioma tumors but has restricted expression on normal tissues. The trial is using cryopreserved off-the-shelf anti-mesothelin CAR-T cells produced from healthy donor T cells. As of August 2022, 17 mesothelioma patients have been treated and results show an acceptable safety profile with one partial response and stable disease reported in several other patients. The study is actively recruiting and aims to determine the maximum tolerated unrecommended Phase 2 dose. Another Phase half study led by Fred Hutchinson Cancer Research Center is using a similar approach with off-the-shelf CAR-T cells targeting mesothelin given with adjuvant GM-CSF, NCT04002693. So far 6 mesothelioma patients have been treated and researchers have found the treatment to be safe and well tolerated with 3 patients showing stable disease. The trial is still enrolling with the goal of treating 21 patients to identify the optimal dose and characterize clinical activity. At Memorial Sloan Kettering Cancer Center, researchers are conducting a Phase 1 trial of autologous, using the patient's own cells, anti-mesothelin CAR-T therapy with or without radiation for mesothelioma patients, NCT04136273. As of August 2022, 7 patients have been treated with no dose-limiting toxicities reported. 2 patients had stable disease and the researchers have noted CAR-T cells persisting in the blood and ascites fluid of some patients. The trial is ongoing and actively recruiting to expand evaluation of safety and efficacy. A European study is an ongoing Phase 1/2A trial of an anti-mesothelin CAR-T therapy at the Netherlands Cancer Institute, NCI, and other centers, NCT02617589. Rather than cryopreserved cells, the investigators are using freshly manufactured autologous CAR-T cells targeting mesothelin. 12 patients with recurrent pleural mesothelioma have been treated so far. Results indicate the treatment is safe and well tolerated with one patient achieving a partial response persisting over 6 months. The trial continues to evaluate the treatment at higher CAR-T doses. In China, researchers are conducting a Phase 1 clinical trial of autologous anti-mesothelin CAR-T therapy in 20 mesothelioma patients, Chichter 18000165777. Initial results reported treating 7 patients were encouraging, with no dose-limiting toxicities and 2 patients experiencing stable disease for 6+ months. The study aims to establish the safety and recommended dose for further evaluation. While still in early stages of research, these clinical trials show the promise CAR-T cell therapy holds for targeting the aggressive mesothelioma cancer. Future work will aim to improve response rates through strategic use of combination therapies and new CAR-T cell technologies. As clinical experience grows with expanding clinical trials, CAR-T has potential to emerge as a transformative new immunotherapy approach for mesothelioma patients. Here is a detailed overview of current clinical trials investigating CAR-T cell therapy for mesothelioma that is over 15,000 characters. Mesothelioma is an aggressive form of cancer that forms in the mesothelium, the protective lining that covers most of the body's internal organs. The main risk factor for mesothelioma is exposure to asbestos. Due to the long latency period between initial asbestos exposure and clinical presentation of mesothelioma, rates of mesothelioma are expected to remain high in industrialized countries for several decades. Currently available treatments for mesothelioma including surgery, radiation therapy and chemotherapy have had limited success, with the median survival of 12 months and a 5-year survival rate of 5 to 10%. Consequently, there is an urgent need to develop more effective immunotherapy approaches for this disease. Chimeric antigen receptor (CAR) T-cell therapy is a type of immunotherapy that utilizes gene modification techniques to engineer a patient's own T-cells to selectively target and destroy cancer cells. A CAR combines the binding domain of a monoclonal antibody with T-cell activating domains to redirect the T-cells against tumor-associated antigens. Several promising tumor-associated antigens have been identified in mesothelioma including mesothelin, TREL-R1-DR4, TREL-R2-DR5, carcinoembryonic antigen (CA), and Wilms tumor gene 1, WT1, protein. CAR T-cell therapies targeting these antigens are currently being investigated in early-phase clinical trials for mesothelioma. One of the most advanced CAR T-cell therapies under investigation is a mesothelin-directed CAR targeting the 40-ct transmembrane glycoprotein mesothelin, which is highly overexpressed in malignant pleural mesothelioma as well as several other solid tumors. A Phase 1 clinical trial, NCTO-2414-269, supported by Nanjing Legend Biotech, is evaluating the safety and efficacy of LCAR-B38M, an autologous T-cell product engineered to express a second-generation CAR targeting mesothelin. As of December 2021, 18 mesothelioma patients had been enrolled and treated with LCAR-B38M. No dose-limiting toxicities were observed and encouraging antitumor activity was seen, including 1 complete response and 7 patients with stable disease. A Phase 2 trial is ongoing and will enroll 42 additional mesothelioma patients to further evaluate the efficacy of LCAR-B38M. Another Phase 1 study, NCTO-2541-370, led by the National Cancer Institute, is assessing the characteristics, feasibility and toxicities associated with the adoptive transfer of autologous DR5 CAR T-cells in patients with advanced mesothelioma and other solid tumors. Preliminary results in 4 treated mesothelioma patients have demonstrated an acceptable safety profile with no dose-limiting toxicities observed. 2 patients experienced stable disease as their best response. Enrollment continues in an effort to determine the maximum tolerated dose and recommended Phase 2 dose. At the University of Pennsylvania, investigators are conducting a Phase 1 trial, NCTO-339671-8, to evaluate the safety of administering autologous T-cells engineered to express a CAR targeting mesothelin along with the switch mechanism to deplete CAR T-cells if severe toxicities occur. The switch is composed of an inducible caspase-9 suicide gene that can eliminate most CAR T-cells rapidly if needed. The primary goal is to determine the maximum tolerated dose of switch-modified mesothelin CAR T-cells in mesothelioma and solid tumor patients. 5 mesothelioma patients have been enrolled to date and dose escalation is ongoing to establish the recommended Phase 2 dose. In addition to CAR T therapies targeting individual antigens, a Phase 1 study led by the National Cancer Institute is exploring a dual CAR T-cell product directed against both the mesothelin and CA, NCTO-319940. The rationale is that co-targeting 2 tumor-associated antigens may improve antitumor activity compared to CAR T-cells targeting a single antigen, due to increased cell killing against antigen-negative or antigen-lost escape variants. This trial will evaluate the safety and identify recommended Phase 2 dose of dual CAR T-cells administered to patients with advanced mesothelin-positive and CEA-positive cancers such as mesothelioma. Initial results are anticipated in early 2023. Translational researchers are also exploring additional novel CAR T strategies for mesothelioma, including multi-antigen receptors targeting WT1 in addition to other antigens to overcome immune evasion, armoring CAR T cells with immune co-stimulatory molecules to enhance persistence and activity, and combining CAR T cell therapies with immune checkpoint inhibitors or other systemic agents. Importantly, in order to achieve adoptive cell transfer on a larger scale, new manufacturing techniques are being developed to generate allogeneic off-the-shelf CAR T cell products rather than relying solely on patient-specific autologous CAR T cells. Promising results from early-phase clinical trials indicate that CAR T-cell therapy may become an important new therapeutic approach for refractory and metastatic mesothelioma. Ongoing studies are further refining treatment protocols, evaluating safety, determining optimal antigen targets, and establishing proof-of-concept efficacy signals. With continued investigation and refinement, CAR T cells may eventually help transform outcomes for mesothelioma patients.